This really is a great honor, and naturally I'd like to start by thanking the Society, in particular the Society members who nominated me for this award, and of course the award committee for this very special accolade. The British Pharmacological Society is a very important society in my career, has played a very important role in my career, not only because it was the first society that I joined, the first society in which, the society in which I gave my first poster and oral presentation, but it absolutely was instrumental in development of my career from a PhD student to an independent researcher. So it's a real pleasure for me to give this lecture, to deliver this lecture in front of so many society members, in front of so many former and present colleagues, and it's a treat for me to have members of my family in the audience as well. So these lectures are traditionally a combination of life stories and highlights of scientific achievements, and as such, I'd like to tell you my story and the career, how I got into the field that I'm in, and the people and the events that impacted my career development. I know this is a relatively mixed audience, but I do hope that everybody in the lecture will enjoy some aspect of this lecture. So just to set the scene, this is what I'm interested in. For almost 30 years or so, I've been asking the basic question, how do white cells cross blood vessel walls? This is a very complex process which plays obviously a key role in our immune response to infections, injury, but of course we know that inappropriate trigger of this reaction can contribute to development of numerous inflammatory conditions as listed here. And the overall objectives of our research and many others is to understand the molecular pathways that mediate this process with the aim of identifying novel targets that could be used for development of anti-inflammatory agents. So what I'd like to tell you in this lecture is how I got into this field, the events and people who impacted my career, and to highlight some of our key achievements, and finish off by where we are and where we're heading. So, to start off, well, inevitable baby images coming up, I'm afraid. I was born in Iran, in the beautiful city of Isfahan, which was actually a capital city of Iran for a number of years, and because of that, has got beautiful monuments such as beautiful bridges, palaces, mosques. And this is me at the age of, I think, 2 possibly. Unfortunately, we didn't stay in Isfahan very long because of my father's job. We traveled almost every 2 years and eventually made the big hop to London when I was about 10. So coming to London, my parents thought that all schools were equally good in London, and we were enrolled, my brothers and I were enrolled in our local comprehensive school, Holland Park School, which I didn't know at the time was actually a very famous school. And the reason it's famous is that it was actually the flagship of the Labour government. It was the first ever purpose-built comprehensive school in London. And in its heyday, it actually had about more than 2,000 pupils. We were actually hated by the local affluent community of Holland Park, but in some quarters, the school was known as the Socialist Eton because of the very strong support of Labour government, and the children of Labour MPs, such as Tony Benn and artists and so on, attended this school. Desperately trying to find a person that this audience would know, or a famous person, I unfortunately came up with this person. He is appropriately one of the most famous Iranians in the UK. So this really highlights the school wasn't very academic, but where it lacked in academic excellence, it was very rich in terms of multicultural, multi-ethnicity. And, for example, when most schools have a symphony orchestra, we had a steel band. And it was actually a very good steel band. I was in it, and we had various tours around other schools. So I was at Holland Park School for 7 years. It was a very interesting time. I learned a lot about being independent, developed resilience, learned to basically get to know people of all nationalities, and these have all helped me throughout my career. Academically, I did okay. I was very good at mathematics, and because of that, I went to do a BSc in mathematics at Imperial College. Unfortunately, I didn't like it at all. I hated it. I left after one term. I found it incredibly dull. I was one of 3 women in a class of 110, but it was still boring. And I basically left and came out, did various jobs, whilst I gave serious thought to my career. Having made a mistake once, I didn't want to do it again. So I considered a variety of courses. I knew I wanted to get back into biological sciences. I missed the laboratory, the experiments. So I considered a range of courses such as biology, biochemistry, physiology, medicinal chemistry, psychology, and I did actually go and visit various departments and talk to various people about these courses. And eventually I discovered pharmacology, and it was absolutely brilliant. This was the course that I wanted to do, and I was really excited when I was offered a position at UCL to study pharmacology. UCL, of course, was the first pharmacology department, I don't know what this message is, hopefully it'll go, first pharmacology department in the country, without doubt one of the most influential pharmacology departments. It was home to giants such as the ones I've listed here, A.J. Clark, Gadam, Schild, Sir James Black, and I had the privilege of being taught by these 3 very eminent pharmacologists, David Colquhoun, Maureen Dale, and Humphrey Wrang. David Colquhoun was my personal tutor. Maureen Dale, I'll talk about her in the next slide, and Humphrey Wrang, our president, of course, was the head of the department when I joined the UCL pharmacology department. I had a fantastic time doing a degree in pharmacology. It was incredibly inspirational doing a degree at UCL, and it really was the firmest grounding you could get in following a career in pharmacology. A number of other events during this period really led me on my career path. I was incredibly fortunate, and I think David Colquhoun actually organized it, for me to have summer jobs at Borough's Wellcome for 2 summers. The first, I was working in the laboratory of Sir James Black, and the project that I was put on was to look at prostacyclin receptors on platelets. This was a new model for Sir James. He was really keen to stay in touch with the data, with the model, and being keen, I used to work quite long hours, and we had a lot of one-to-one discussions, and I couldn't get away with anything. You can imagine discussing agonists, partial agonists, antagonists with Sir James Black at any level. It's pretty daunting. As an undergraduate, it was pretty terrifying. But he was an amazing teacher, very generous with his time, wicked sense of humor, and it was absolutely one of my greatest inspirations to follow a career in pharmacology. So that was incredibly enjoyable summer. Because of the prostacyclin aspect, I decided the following year to exchange groupings and broaden my skills, and I joined the group of prostaglandin research where Rod Flower and Brendan Whittle were very much looking after me. And collectively, these really enhanced my passion for pharmacology, and in particular immunopharmacology. And when I went back to university the final year, we had this wonderful immunopharmacology course run by Maureen Dale, and I took this and I did a project with her on neutrophils, and it really set me off on the neutrophil path. And I think it was actually wonderful that 30 years after Maureen Dale started me on this career path, we could actually contribute to one of the covers of her famous book. This was the 7th edition, and this was an image taken by one of my fellows, Abigail Woodfin. So that was very gratifying to be able to contribute to that after 30 years. So having been started on an immunopharmacology course, on a pharmacology path, I wanted to follow this up and decided to do a PhD in immunopharmacology. And I joined Robin Holt's laboratory at King's College. Robin was an eminent immunopharmacologist, did a lot of work on prostaglandins, and this was a really enjoyable time as well. This is a picture that I'm grateful to Jude Hall and Ian Morton, and this was, as well as great science and academics, it was a very supportive department. People like Ian Morton, John Halliday, this is Robin. They just acted as mentors to all the PhD students, and it really was a very enjoyable time. And Gus Bourne was the head of the department at the time. So Robin, sadly, this is a picture again Jude Hall gave me, sadly passed away 15 years ago, and this is a picture from one of our very famous Christmas parties, and they were always fancy dress. Robin, I'm indebted to him at a number of levels. The first is that when I joined his department, he basically said, "What would you like to work on? What excites you?" I said, "Well, I've done some work on neutrophils, so I quite like to follow on neutrophils if possible." And he said, "Well, okay, we'll put you on a neutrophil project." So that was the first actual neutrophil project in his laboratory. That's the title of my thesis, and it meant that I had to set up all the assays, do a lot of the background reading, but he was always there to support and guide, of course. But I didn't appreciate it at the time, but that really was fundamental in developing me as an independent scientist. The other thing that I'm very grateful to Robin is that he introduced me to the British Pharmacological Society. We presented posters, oral communications, and it really was a forum for developing and meeting other pharmacologists. And, of course, I'm grateful to him that he was a reasonably successful PhD, and we got a number of papers out of it. Now, why was I interested in neutrophils? Well, this was a movie that I think I saw first time during my immunopharmacology module with Maureen Dale, and this really grabbed me, and I'm sure a lot of you have seen this. It's a very famous movie, and it shows a neutrophil chasing a bacteria on a glass cover. And this, even non-experts, when they see this, and I've shown this to my family, they're immediately stunned as to how is this neutrophil know where this bacteria is? How can it chase it? And now it's gobbled it up. How does it know where to go? And how does it coordinate this movement so exquisitely? And this, of course, comes down to two principal functions of neutrophils: their ability to exhibit directional movement, chemotaxis, which is mediated by chemoattractants, and their ability to adhere and migrate, mediated by adhesion molecules. And these are the two areas that I've gone on to really investigate all throughout my career. So neutrophil migration is, of course, a fundamental response to infection and injury. Neutrophils are the first cells to arrive at sites of infection, and this, of course, is a characteristic response of an inflammatory reaction. We can now image this response very elegantly using technique of intravital microscopy, and this now shows neutrophils coming out of an inflamed blood vessel using the mouse cremaster muscle. And this technique has also played an important role in my career. I think the movie that I showed you earlier of a neutrophil chasing a bacteria really demonstrated to me the power of imaging, and I went on to really be passionate about visualizing events, inflammatory events. I would like to highlight that this technique is not a new technique, and although it's very elegant, the first observations of leukocyte vessel wall interactions that I'm aware of were reported by Augustus Waller, who's a British physiologist, and this is an example of beautiful hand-drawn images that he published. And the beautiful thing about these works, these early works, is they also eloquently described their responses, and this is a phrase that Augustus used. He described adherent corpuscles as he compared them to pebbles or marbles over which a stream runs without disturbing them. And I cannot express the response any better than that. This is absolutely what we see. Following on from Augustus, Julius Konheim, of course, the German pathologist, he made huge progress in the inflammation field, a lot of it through imaging. And I very much like this phrase, and I think it's actually humbling that he said this so many years ago, that the underlying mechanism of leukocyte migration is mediated by molecular changes in the vessel wall. This is exactly what happens, and this is exactly what we are investigating even today, trying to understand these molecular changes. In the modern era, the work of Gus Born was absolutely fundamental to this area. Together with his colleague Anne Atherton, they absolutely determined or set the bar for quantitative analysis of leukocyte vessel wall interactions, really investigating in a lot of detail the relationship between the velocity of granulocytes in relation to blood flow. And Gus, of course, was head of my department at King's College when I did my PhD, and I interacted with some of his colleagues, and I was fascinated with some of these images I saw even then. Again, a very profound statement in the discussion of one of their papers, that the increased adhesion forces must depend on structural changes in the molecules which determine the interaction of granulocytes with the endothelium. At that time, these were entirely unknown. So again, very profound statement, and this, the identity of these molecules was discovered a number of years later, led by Tim Springer and colleagues. So we knew that neutrophils can interact with endothelial cells, but the molecular players were not known, and Tim Springer and colleagues, they identified the CD11, CD18 antigen complex, beta-2 integrins expressed on neutrophils, and these are constitutively expressed, and they need to be activated by chemoattractants such as chemokines. So Gus Born's hypothesis was absolutely correct in that there has to be molecular changes in these molecules. And Tim Springer and colleagues went on to identify the ligands for these structures on the endothelial cells as being ICAM1 and ICAM2. So this was a huge step in our understanding of the molecular events that mediate leukocyte vessel wall interactions. And it also explained the underlying cause of this disorder, leukocyte adhesion deficiency, where the patients are highly immunocompromised. They had defective neutrophil adhesion and phagocytosis, and it was found that the cause was due to defective expression of beta-2 integrins. So these events were happening whilst I was doing my PhD, and in order to investigate these events in vivo and learn more about inflammation, I went on to join the MRC Clinical Research Center at Harrow, and this was the section I joined, section of vascular biology, headed by John Gordon. It was absolutely at that time one of the leading, most innovative vascular biology groupings in the country. There was two branches to it, the endothelial cell group and the inflammation group. Endothelial cell group was headed by Jeremy Pearson, who is an emeritus professor of vascular biology at King's and also the BHF medical director. And Jeremy has remained a great friend and mentor all throughout my career. I joined Tim Williams' group, who without doubt was one of the leading inflammation biologists and has made huge contributions. And all throughout my career, I cite him. I've learned much of what I know about inflammation from Tim, and I'm grateful to him. I joined the team. It included Susan Brain, Peter Jost, Mark Rampart, Paul Helliwell. These were the A-team of inflammation biology, and I was really delighted to be part of them. It was very exciting, and again, these guys have stayed great friends and mentors all throughout my career. So we started to investigate neutrophil vessel wall interactions. This was the scheme, and we wanted to investigate what happens when you block these molecules, looking at various inflammatory models in vivo. So we collaborated with John Harlan at University of Washington. He had antibody to the beta-2 integrins, monoclonal antibody 60.3, and we investigated this in various inflammatory models, the arthritis reaction, zymosan-induced neutrophil accumulation, and a passive cutaneous anaphylactic reaction in rabbits. And you can see that in all of these reactions, treatment of neutrophils with this antibody, monoclonal antibody 60.3, suppressed accumulation of neutrophils in rabbit skin. This antibody was considered for numerous clinical trials, in particular hypovolemic shock, but it was unsuccessful. But similar antibodies such as anti-LFA1 blocker, which is LFA1 being another subunit of the beta-2 integrins, another component of the beta-2 integrin complex, it was used for psoriasis, but again, due to adverse effects, it was withdrawn. So at that time, we were beginning to now learn quite a lot about the leukocyte adhesion cascade. This is the emergence of it. The selectin molecules were identified as molecules mediating rolling of leukocytes, and then the integrins, the firm attachment, and we knew that eventually leukocytes began to migrate out of the vessel wall, as shown here. What was missing was the activation. What molecules were actually stimulating the activation of leukocytes, and they're sticking to the vessel wall. And a major contribution to the field was discovery of the chemokine family, led by people such as Yoshimura, Schroeder, Waltz, and colleagues. And of course, Tim Williams and others, including John Westwood, they made huge contributions to the biology of the chemokine family. And this is a family which we now know exhibits numerous biological functions, including metastasis. They have roles in angiogenesis and so on. But they were very much identified based on their ability to stimulate leukocyte migration. And this is very clearly illustrated here. This is human neutrophils before and after application of interleukin-8. And you can see this rapid spreading of neutrophils on glass slides. So we went on to study some of the activities of IL-8, and the field was progressing fast. We knew that in response to an inflammatory trigger, such as infections, allergens, tumors, autoantibodies, chemokines were generated. They generally were expressed on heparan sulfate proteoglycans on the surface of endothelial cells, and this promoted their interaction with receptors on leukocytes, largely GPCRs, G protein coupled, and this promoted activation of integrins, which then supported the adhesion and migration of leukocytes out of the vessel wall. And together with Tim, we did some studies of IL-8-induced neutrophil migration. In particular, one of our studies I thought was very interesting in that we looked at the role of G protein-coupled receptors in this phenomena, in that we showed that pretreatment of indium-labeled neutrophils with pertussis toxin in vitro inhibited their accumulation in vivo in response to intradermally injected interleukin-8 in rabbit skin. And this, I think, was the first demonstration of G protein-coupled receptor-mediating chemokine response in vivo. So by then, Tim had been appointed as a professor of applied pharmacology at the National Heart and Lung Institute, and a number of us went with him. I was appointed as a lecturer on relocating to NHLI, and this was a good period in that Tim supported me to apply for a Wellcome Trust fellowship, and this led on to becoming a senior lecturer and a Wellcome Trust University Award, which really put me on a tenure track at NHLI. As well as Tim, I need to acknowledge two other people in this period. Tim Clark, who was the head of NHLI at that point, and Marion Kimberley, who was the administrative dean, and they were very supportive of my application for Wellcome Trust University Award. During this period, another major achievement was the arrival of these guys, who obviously brought a lot of joy, but also caused a lot of havoc and complexities in the career as well. But absolutely something I would recommend to everybody. So I went on to study asthma and allergy because that's what we were focusing on. So there were two branches to our investigations of mechanisms of eosinophil migration, identifying the molecules that attract the eosinophils into sites of inflammation, and investigating the mechanisms that mediate eosinophil migration. In terms of the eosinophil attractions, this was a hugely successful arm of the research, the department's research, led by Tim and Peter Joes, and in that they identified eotaxin, which was very much it was the most potent eosinophil chemotactant chemokine, and this went on to really excite the whole field in terms of drug development possibilities. In terms of adhesion, I was involved in developing techniques and investigating the mechanisms of eosinophil adhesion. And together with Lucia Faccioli, a postdoc from Brazil, we developed a model for looking at eosinophil migration in vivo. So what did we know about eosinophil migration at that time? Well, it was, again, a very dynamic stage of the field. We knew about neutrophils. I've already mentioned the CD11, CD18 antigen complex. But we knew very little about migration of other leukocyte types, and in particular about eosinophils. And this changed with the discovery of the VLA proteins by Martin Hemmler and colleagues in Boston. And in particular, for eosinophils, the molecule VLA4 was found to be instrumental in their adhesion. What was their ligand on endothelial cells? Roy Lobb at Biogen cloned this molecule, VCAM, vascular cell adhesion molecule, and this really started developing as a very exciting avenue for investigations of allergy and inflammation. What wasn't known at that time was the role of this pathway in eosinophil accumulation in vivo, and we wanted to explore this, so we collaborated with Roy Lobb, who cloned VCAM1, as I said, from Biogen, and he provided us with tools to do this. He had antibodies against VLA4, which we could use that cross-reacted with our animal models, and we tested this anti-VLA4 antibody, for example, in a model of cutaneous anaphylactic reaction in the guinea pig skin, and you can see there was very significant suppression of the eosinophil migration. This was the first in vivo evidence for involvement of this molecule in eosinophil migration in vivo. We went on to look at this pathway in numerous other inflammatory reactions, and there was a series of quite important papers came out of it. One person who made a lot of contribution to this field, to this aspect of our work, Maria Sanz, I want to acknowledge her because she went on to become a very eminent pharmacologist back in Spain, where she was from. She joined us for about 3 or 4 years, and she's now a successful professor of pharmacology at University of Valencia. And I was delighted to hear recently that she's actually been elected as president of the Spanish Society of Pharmacology, which is quite an achievement in Spain. It is incredibly hard for women in Spain. So Maria made huge contributions to these works. So VLA4 blockers, they were incredibly exciting, and they were used based on some of it was based on our works actually. They were tried in trials of asthma, but unfortunately they were unsuccessful, possibly because a cohort of patients just weren't selected properly. But they are currently in clinic for use in multiple sclerosis. So there is success in the story of VLA4 blockers. So whilst we were doing these works, I was involved in doing some imaging of eosinophil migration across blood vessel walls. And again, this really caught my interest, and I wanted to really investigate. I decided analyzing leukocyte migration across blood vessel walls was something which needed to be done, and we knew very little about it. So how do leukocytes cross blood vessel walls? Well, they have to cross endothelial cells. and they have to cross associated pericytes and the basement membrane layer. These events were really largely, up until that point, investigated only by EM analysis. And this is an example of one of our own EMs, where you can see a neutrophil sending a protrusion through an endothelial cell. We knew almost nothing about the molecular events. This was a complete black box. We knew, of course, that there had to be some molecular structures between endothelial cells, such as shown here, but the nature of these molecules was really unknown. So the major breakthrough here was the discovery of this molecule, PCAM1. Bill Muller first identified the expression of this molecule using antibodies that he generated against proteins expressed at endothelial cell junctions. He then went on with Peter Newman to clone it, and Steve Albelda developed the story even more. What was very exciting is that Bill Muller then showed that this molecule is very much involved in migration of leukocytes through endothelial cell junctions. And at that time, I was following the field very carefully, and I was very excited when Steve Albelda published a paper in Science showing, again, that PCAM1 was involved in neutrophil migration, this time in vivo, because up until that point, it was all very much in vitro studies. So for the first time, there were antibodies available that could be used in vivo models. And I immediately contacted Steve Albelda, and he was very happy to collaborate with us, and he sent us some of his antibodies, and we tested them by intravital microscopy. And this is a technique that I really got quite excited about initially from working in Gus Bourne's department, but subsequently when I was working with Tim, of course. So we tested the anti-PCAM antibody on neutrophil migration in rat mesenteric vasculature, and what we found very interesting was that this antibody was not inhibiting neutrophil migration at the level of the endothelium, but it was inhibiting neutrophil migration at the level of the basement membrane. We saw this lining of neutrophils at the level of the vessel wall, but not inside the lumen. And EM analysis clearly showed that there was a lot of capture of neutrophils in the vessel wall at the level of the basement membrane. So this was the first evidence that PCAM1 was mediating neutrophil migration beyond the endothelial layer. And we went on to investigate, and this work was led by a PhD student, Matthew Wakelin. We went on to investigate PCAM deficient mice a number of years later, and this was Richard Thompson, who's a clinical research fellow working with me who did all this work. And what Richard found was that we could inhibit neutrophil migration in these PCAM knockout mice in response to IL-1 beta, but not TNF-alpha. So a stimulus-specific role for PCAM1 was being identified. But again, we found that this inhibition was at the level of the basement membrane. So collectively, using both an antibody blockade approach or a knockout approach, we have evidence that PCAM1 was involved in neutrophil migration in a stimulus-specific manner in vivo, and that the PCAM1 was very much involved in neutrophil migration through the venular basement membrane. So how was this working? Well, we had to obviously investigate the mechanism. It was unexpected. And what we found was that basically PCAM ligation during neutrophil migration through the endothelium, so this is PCAM on endothelial cells interacting with PCAM on neutrophils, as neutrophils are moving through the endothelial cell junction, leads to upregulation of this integrin alpha-6 beta-1. And this integrin alpha-6 beta-1 is a very important receptor for components of the venular basement membrane. So this was the mechanism that we offered for how PCAM1 was involved in migration through the basement membrane. And this is a very nice image that shows that as neutrophils migrate across blood vessel walls, this is the neutrophil inside a blood vessel, and these are neutrophils outside, the expression of alpha-6 beta-1 is upregulated on their surface. So this really demonstrates that this triggering of alpha-6 expression was regulated at the level of the vessel wall. And this was actually an example of how transmigration can regulate the phenotype of emigrated neutrophils, which really primes them for an effective immune response once they get into the tissue. And I don't have time to tell you about some of the other works in this aspect. So around this time, I left the Brompton campus of NHLI and joined the Hammersmith campus, and here I joined the team headed by Dorian Haskard, the Unit of Cardiovascular Medicine. This again was a very, I think, productive period. I was there for about 10 years. Largely, I think, not largely, but it's certainly contributing. We were right next to Wormwood Scrub Prison, which meant that there was no distraction. We could just get on with the work, no leaving the lab. So this led to me becoming a reader, and I was awarded a personal chair at this period. And we really focused on the mechanisms of transmigration, systematically looking at all these stages, the endothelial cells, the pericytes, and the basement membrane. An important aspect of our work here was to really enhance our imaging capability, and these two fellows, postdocs, joined my lab, Abigail Woodfin and Mathieu Benoit Voisine, and these guys were incredibly talented and excited to do imaging, and both of them, I'm delighted to say, are now on independent fellowships and doing their own research. This is the kind of imaging we started to establish to really look at localization of neutrophils in relation to different components of blood vessels in 3D, and we could really investigate the involvement of specific adhesion molecules in this process. So using this technique, we went on to investigate the endothelial cell component, and at that time, a lot was being discovered about endothelial cell junctional molecules. In addition to PCAM1 that I've mentioned, other molecules were identified and cloned, and we collaborated with numerous people around the world to investigate these. We obtained specific blockers, antibodies, use of genetically modified mice, and a lot of imaging to investigate these molecules. And collectively, our work really identified that endothelial cell junctional molecules can act in sequence and in a stimulus-specific manner to mediate neutrophil transmigration through blood vessel walls. For example, we found ICAM2 was very much involved at the early stage where neutrophils are just finding the junctions. Once they're in the junctions, other molecules such as JAM-A take over, and I've already told you that PCAM1 is very much involved in crossing blood vessel basement membrane. So in this context, in this aspect of the work, we know a lot about the molecules that are involved. What is really challenging is to investigate the role of multiple molecules, and I've already mentioned we have indications about their roles in stimulus-specific reactions, and there is a lot of ongoing works now to investigate their roles in terms of regulation of migration of specific leukocyte types or in different vascular beds or in different stages of transmigration. One molecule that we've spent a lot of time investigating was this molecule, JAMC. And this molecule was cloned by Beat Imhoff, who's a great friend and has been a wonderful collaborator. And this molecule has really, it hasn't ceased to surprise us. We've been investigating for a number of years, and it's proved to be a very interesting molecule. Very briefly, it's a member of a larger superfamily of molecules as listed here. It can interact with itself as well as other members of the JAM family and the integrin MAC1. It's expressed on leukocytes, platelets, and endothelial cells, and this expression profile really accounts for its functions as listed here. So Beat and I were collaborating for some time, and we decided we needed to put more emphasis on this work. So we put a joint grant in, and we got a PhD student, Christoph Scheiman, joined us on this PhD student application, and he was really looking at the role of JAMC. Initially, we were looking at inflammatory models, because that's what we're interested in, and he looked at models of ischemia-reperfusion injury and neutrophil migration, and looked at JAMC knockout mice, and he found that there was an inhibition of neutrophil migration in a model of ischemia-reperfusion injury. Whilst Christoph was doing this work, he came across an unexpected expression profile of JAMC. So this is a PCAM labeled blood vessel. This is JAMC expression. So although it's expressed at low levels on blood vessels, there was a very strong expression somewhere else. And Christoph came to me and said, look, I can see this expressed on some other structure, but I don't know what it is. A little bit of extra work, we found that Christoph had identified JAMC expression on myelinated peripheral nerves. This was completely new, unexpected, and as we did more and more work, the evidence became stronger and stronger. And I was in a position of uncertainty. What do we do with this? I'm not a neurobiologist. We don't know how to take this work forward. Should we just put it in as an appendix in one of our usual inflammation papers? But he was just so excited by this, and I was driven by his excitement. And this is a real lesson to PhD students. Don't give up anything. Do try and train your supervisors if you find something exciting. And on the back of his excitement, we really went on. We forged collaborations with neurobiologists, neuropathologists, electrophysiologists. And I remember actually I had a brief conversation with Humphrey Rang. I wanted his advice on people I could talk to. And basically we took this work forward. We found that JAMC was expressed on junctions in myelin folds around peripheral nerves, so expressed as Schmidt-Lantermann incisures and inner and outer mesoaxons. And we had JAMC knockout mice. We showed that they had abnormal morphology and integrity of peripheral nerves, defective nerve conduction, defective motor behavior. And I think the icing on the cake was our collaboration with Pravin Anand, where we found that JAMC is also expressed in human nerves, and very excitingly, it was reduced expression in nerves from patients with peripheral neuropathy. And I'm really proud that we managed to get a science paper for Christoph on this work, and this was absolutely the platform for him to go on to get a fantastic fellowship to go to New York, and he's now a PI in Munich. So, in terms of these other aspects, coming back to our areas closer to our hearts, crossing the pericytes and the basement membrane, our work has also really identified some unexpected avenues. The basement membrane, of course, is a formidable barrier to emigrating leukocytes formed by laminins and collagen type 4, interconnected by various other proteoglycans, and we know that neutrophils really stop at this level for a significant amount of time. So the question is, how do leukocytes cross this barrier without disrupting its intricate and critical structure? And we identified, this is the work of Shijun Wong and Mathieu Benoit Boisine, by imaging again, that the basement membrane had these permissive regions, these regions that exhibited lower deposition of certain basement membrane proteins. And these regions are absolutely aligned with gaps in the pericyte layer, and we know that they are the preferred sites of leukocyte migration, and they seem to support leukocyte migration through regulated remodeling and possibly access of extravascularly generated directional cues. May also have a pathological role in that excessive or prolonged enlargement of these molecules may lead to inappropriate leukocyte infiltration and compromised vascular integrity. And I think a very exciting possibility is that they may actually be involved in tumor intravasation as well. So around this time, a very complete by chance meeting again, brought Flower in an airport lounge. This is absolutely true. Both of us were flying to Canada. We bumped into each other. Both our flights were delayed due to weather, I think. And Rod invited me to come and visit William Harvey Research Institute. And before I knew it, I was having a conversation with Mark Caulfield and tea with head of the medical school, Nick Wright. And this all happened very fast. And I'm grateful that these guys made me an interesting offer, which I haven't regretted at all. It's been one of the best moves of my career to move to the William Harvey Research Institute, which is, of course, one of the great pharmacology institutes in the country and beyond. And I want to acknowledge all my colleagues within the Institute, Medical School and QMUL for supporting myself and my team in a very smooth transition into the Institute, in particular Mauro Peretti, who was deputy head of the Institute at that time and is now co-director. So this is us moving in. I've moved in with 3 people, Abigail, Mathieu, and Christophe, and the team grew rapidly since then, and this is us last year. And what we've been allowed to do is to develop a grouping to investigating microcirculation. So our interest is now broadened. Of course, microcirculation is involved in oxygen delivery, maintaining vascular tone, and primary cytol, where an inflammatory response is mounted. But of course, you can also get defective, abnormal microcirculation responses, which can contribute to numerous pathologies, such as listed here. And we have interest in a broad range of these activities. My own interest has focused on leukocyte vessel wall interactions, and at this stage, 2007, we knew quite a lot now about the molecular pathways, the signaling pathways associated with them, and I was privileged to be involved in writing this review with Klaus Ley, which has now been cited over about 2,000 times, and this really sets the scene as to what we know even now about this process. What we wanted to do, I saw it as an opportunity when we moved to the William Harvey to really again enhance our imaging capability for addressing more fundamental questions, and we wanted to investigate this process in more detail, in particular the dynamics of this process. We knew that breaching a vessel wall can take up to 30 minutes or so, but why did it take so long? What were the neutrophils doing in this period? So we set up a 4D imaging platform. This is 3D in real time. Again, this was led by Abigail Woodfin. And the kind of images that we've acquired have been phenomenal, and they really have set the bar for many other groups, and we can really track neutrophils going through blood vessel walls in quite incredible detail. This is a neutrophil going through a PCAM-labeled endothelial cell junction, and we can really take this forward now to investigating phenomena beyond the endothelium. So this is looking at neutrophil crossing the endothelium and crawling within the vessel wall now, and this really told us what the neutrophils were doing for this 30 minutes lag time. There was a lot of subendothelial cell crawling going on, and we started looking at this phenomena in relation to pericytes using other reporter mice. This is a mouse that has got RFP expressed on pericytes and smooth muscle cells, and you can see that this subendothelial cell crawling is very much supported by these pericytes, and we now know that pericytes express adhesion molecules and chemokines, and this is instrumental in this crawling response. And this is work that was led by Doris, who's a PhD student, with a lot of help from Mathieu. So we have a lot of handle on the mechanisms, dynamics of leukocyte migration through the endothelium. This is a rapid response. Then we know there's a lot of subendothelial cell motility, and then the neutrophils need to cross the basement membrane as mediated by these permissive sites. Very quickly, what we found as part of this work more recently is that chemoattractants can also cause shape change in pericytes, and this was work done by another PhD student, Mickey. And what we found was that this pericyte-induced shape change, as induced by chemoattractants, was completely neutrophil dependent, so absent in neutrophil-depleted animals. And also, interestingly, it was totally dependent on TNF receptors. So, cutting a long story short, we found evidence for endogenous, what we believe to be neutrophil-derived TNF, acting very close to the vessel wall in response to chemoattractants. What was the functional role? And this is basically summarizes this finding, which was published this year, and that we found that neutrophil-derived TNF plays a key role in mediating neutrophil-mediated microvascular leakage. So chemoattractants acting on neutrophils induce rapid release of TNF that act on TNF receptors, and this causes increased vascular permeability. Of course, this phenomena was reported by Tim Williams with Caroline Wedmore in 1981, but the mechanism wasn't really known, and we were delighted that Tim was involved in this study. And of course, there's significant interest in the use of TNF blockers in numerous inflammatory conditions characterized by excessive edema, and we believe that our observations could contribute to a broader use of TNF blockers and better understanding of how they're acting. So just to finish off, this is another exciting avenue that we are now following, in that Abby, as part of her initial investigations of this model, made the observation that neutrophils can also exhibit reverse motility. So this neutrophil is crossing endothelial cell junction and then exhibiting a reverse motility back into the circulation. And this phenomena being shown again. This is a neutrophil in the subendothelial cell space, and then it's crawling back out into circulation. So this is a abluminal to luminal motility of neutrophils. It was totally unexpected, and we were very keen to investigate the frequency, mechanism, pathophysiological relevance of this. And basically what we found, again, it comes down to this molecule, JAMC. It plays a key role in regulating the normal migration of neutrophils from the lumen to the ablumen. When JAMC is lost from endothelial cell junctions, such as reactions caused by ischemia perfusion injury, neutrophils lose their directionality and they exhibit reverse motility. And this is the model that we're working on. We believe that reverse transmigrated neutrophils, they're activated and they can enter other organs and cause distant organ damage. And this is a new paradigm in terms of how a local inflammatory response can become systemic. And this, of course, has implications to numerous human conditions such as trauma, ischemia perfusion injury, post-surgery susceptibility to SIRS. And we are collaborating with trauma clinicians in particular, as this work has huge translational potential. So what it tells us really is that it's no longer sufficient to quantify neutrophil migration at sites of inflammation. You need to know how they got there, the dynamics of the process. And so we're following this up very aggressively in terms of understanding the mechanisms of this process, the pathogenic potential of reverse transmigrated neutrophils, and if we block this, can we protect distant organ damage, and also other aberrant modes of leukocyte transmigration. And we've made quite a lot of progress on these fronts. So just to finish off, we've moved a long way in terms of the adhesion cascade. We understand the intricacies of this process. We know what triggers the reaction. And in terms of drug development, some progress has been made in that this drug is an alpha-4 blocker, as I mentioned, is in use in treatment of multiple sclerosis and Crohn's. A new generation of this drug, which is more gut-specific, is being used in treatment of ulcerative colitis and Crohn's again, and other indications are in the pipeline. Progress has been slow, and we do need to make greater advancements, and we need to work on understanding the intricacies of these events, both at the cellular, subcellular level. We need to use more advanced, more complex inflammatory models, such as impact of cellular senescence. We need to understand the broad role of white cells, in particular neutrophils, in acute and chronic conditions. And I'm convinced that we will identify disease-specific phenomena with the technology which we have at hand at present. So this is the end of the science. I want to move on to a few acknowledgements. It's very difficult to wrap a lot of people who've impacted my career in a single slide. I've mentioned a number of people already, But in putting my acknowledgement slides together, I consider 3 groupings of people who've really impacted me. One have been the fact that I've had the opportunity to work with formidable women in my career. And I've learned so much from these ladies. Some I've mentioned already. Others have been recipients of this award. Julia Buckingham has been a constant mentor and role model. Many others that I don't have time to talk about. And I think there are challenges for women in any profession, and it just makes it so much easier when you can discuss with other women who've been there. So this is why I think the BPS mentoring scheme is such an important scheme, and absolutely, I've been a keen follower of that. Secondly, I've been incredibly lucky to work with many talented young people. About 30 or so PhD postdocs have passed through our labs. Many of them are successful in their own rights now, independent researchers. I've mentioned some of them, and this is the current team that I want to acknowledge for putting microvascular research on the map, and we're doing some really exciting work. Of course, we're indebted to our funding bodies. In particular, I need to acknowledge Wellcome Trust for supporting me all throughout my career. And finally, you need very strong family support. It's incredibly hard, and this is not an easy. This is not a 9-to-5 job, as you all know. This is me and my 3 brothers when we came to this country, and this is us enjoying the party after my inaugural lecture. You can see how happy they are. They're incredibly proud of everything I've achieved, and these are my parents who really have helped me hugely in my career. And I want to acknowledge my family, my immediate family, my 2 sons and my husband who are sitting here, for the support they've given me, for putting up with endless deadlines, travel commitments that you all understand, and have really helped me follow this career that I absolutely love and I'm passionate about. And finally, thank you for your attention.